going to tell you stories about 5 different papers that we've worked on over the years. So in my research group, I am interested in what controls host-associated microbial diversity. I'll be talking to you about the mammals and the milk microbiomes, but I also study skin microbiomes of amphibians in the context of disease. We also are working in soil microbiomes and sustainable agriculture, trying to figure out how to get farmers to farm more sustainably to increase biodiversity. And then we're working on mammal microbiomes, which I've really expanded on as I came to work at the National Zoo, and thinking about how we can improve their health for the animals that are in our care for reintroductions, and just keeping species healthy. So why mammalian microbiomes? So as you all know, many species are threatened with extinction. I mean, here at the zoo and zoos across the United States and Canada, there's many that are in human care that we are sustaining to have genetic diversity and to focus on reintroductions in some cases. And the more I've worked here, the more I've learned that there are just these persistent problems in some species and some individuals that suffer from GI issues. It's similar that we kind of have this modernization of the microbiome for these animals in captivity, and they also get inflammatory bowel disorder and gastritis. And we're really starting to think more about how this may stem from maternal effects, that the mother may have an unhealthy gut. During nursing, she may actually seed her offspring with an unhealthy gut microbiome, which then facilitates disease and challenges with reproduction and sustaining these populations. In the case of milk microbiomes, we have this really unique data set or collection of samples. We have over 16,000 samples from over 200 species. And, if you guys ever have any interest in these, feel free to reach out. They really offer unique perspectives about the evolution of milk. And many of them have nutrient data. And the primary reason why we have these milk samples is for when mothers reject their offspring in captivity, and we need to come up with a formula. And so every mammal species has a unique combination of protein, sugars, fats, water, and we need to make that the right one. And one of the big famous ones was feeding Fiona was a big one that, Washington Post and a whole bunch of different news articles covered about how we had a hippo sample and we figured out what the right cocktail was for the formula. But I want to, before I get into the story, I just wanted to give special thanks to a couple integral team members here. So Mike Power, he's the milk nutrition expert. He's been my colleague in this from the beginning and is the curator of the milk repository at the National Zoo. Sally Bornbush has been a postdoc working with us for a few years. She's a mammalian microbiome specialist. She's really interested in how you can use FMTs, probiotics, prebiotics to really help those animals that are in human care. And we also had a postmaster student, Mia Katie, who worked with us for a little over a year working on the milk microbiome samples that we had. And I will give a special shout out to Sally. She makes all the incredible graphics. If the graphics in my talk are really poor, I made them. And if they're amazing, Sally made them. So this is an example of one of the graphics that Sally made for a review we recently did. And as you all know, milk is, and its microbiome is an ancient adaptation that unites all mammals. Every mammal produces milk. Every mammal that we have looked into has a microbiome. And it's a really complex story about where do those milk microbes come from, right? So there's evidence for this the oral mammary trafficking pathway. It's a recent one that people have found evidence for, which is really fascinating. I think the enteromammary trafficking pathway, it's been discussed for a couple decades now, where there's some evidence in human and mice models that gut microbes from the maternal gut can get on dendritic cells, be brought through the lymph nodes into the bloodstream, and then into the mammary gland. And I'm really particularly interested in this pathway because if we can change the diet of the mom, we can then impact the milk microbiome and then impact the health of the offspring. So I think the enteromammary trafficking pathway is one of those that may be more easy to manipulate. But you also have mom skin, baby skin, the oral microbiota. I think you all are fairly well attuned to all of these kind of varieties of ways for which the mammary gland and the milk microbiome could be seeded. But then once you have that milk microbiome and it is going into the infant, right? It's one of these first early colonizers of the gut microbiota. The new cell paper, right? It's training the immune system. It can be affecting asthma, nutrition, metabolism, and we believe it applies to all mammals across the board. Okay, so that's kind of the backdrop to the story of the mammalian milk microbiome. And so what can control this microbial community? And we can go way in ecological theory and think about deterministic and stochastic processes. So you have a niche for which can be filled by specific organisms, or the communities could just be assembling stochastically. So they could just be these neutral processes. And so in community ecology and ecological theory, there's selection, dispersal, diversification, and drift. And where selection is the most deterministic, dispersal is how microbes can move from environment to environment, is generally more stochastic. Diversification is just the evolution of these microbes over time, which is also inherently more stochastic. And then just ecological drift, 100% stochastic. And so we've focused on kind of generating this list of what could be these selective pressures that could be what is causing selection and what we can actually manipulate or think about to explain why we see the microbes. We have host diet, evolutionary history of the host environment, the nutritional content of the milk, and infant age. And so we have had 5 published papers to date. The first one was in 2019, and the last one just a few months ago. And I'm going to try to synthesize all of them to kind of tell a consistent story of what we've been learning. So we quantified the community of bacteria in milk. So this is what I'm generally referring to as the milk microbiome of 87 primate mothers. This was our first paper from 9 species. And when I'm saying the bacterial community, we're quantifying the bacterial DNA in the milk. And for us, we are specifically looking at the 16S rRNA gene. So we're just using one gene to figure out what the microbes are. We are collaborating on projects doing shotgun metagenomics and also looking at gene expression as well, but they're in analysis stages right now. And I don't know your guys' background, so just to give you a little bit of an idea of some of the things that I'm going to be talking about, I'm going to be talking about alpha diversity. So we're calling these bacterial ASVs, so amplicon sequence variants, which you can kind of think of strain level bacteria. And so we can sequence these. This is my terrible graphic, where this is a sample of milk. These are the 6 different bacteria species that are there, represented by different nucleotides. And when we find one nucleotide different, we call it a different strain. Where this mantle howler monkey can have 5 different strains, where the human could have 6 different strains. And then we're also interested in this community sharing. And this is beta diversity, the next level, and figuring out how many of those bacterial species are actually shared. And so this overlap, so for instance, humans and mantled howler monkeys may share more of a community, and so they have a higher beta diversity score versus the gorilla and humans, which may share less. And then we can take that and we can put it into a plot, principal coordinate analysis, where if you're more similar to one another, you have a more, so the human and the mantled howler monkey was more similar in this example, and so their points on the plot are going to be more close together than that to the gorilla. Okay, so then let's start with what this first study, what we found. So we found that there were 50 to 600 bacterial strains that exist in primate milk, with humans, howler monkeys, and macaques being most distinct. So here we had, in this data set, was 28 humans, 13 mothers from the urban part of the Philippines, 13 mothers from a rural section of the Philippines, and 2 women from the US here in the DC area that had donated to the milk repository. So lowest microbial diversity compared to all of our primate cousins. Mantled howler monkeys were then the next lowest, and they are actually unique here. They're the only other species here in this data set which are in their natural settings. All the other species were actually animals in captive facilities, while these were sampled in the wild. Just anecdotally, I don't know how that if that has anything to do with this pattern. And then when you're looking at the community composition, you see that humans are quite distinct, macaques very distinct and closer to the howler monkeys. And then many of the other species kind of clustered together, but we really didn't have the greatest replication to ask more resolved questions about their composition. So we had for 6 species the nutrient composition of the milk. We had sugar, fat, and protein, so the general energy that was coming from each of those nutrient profiles. Sugar was not significant in this model, but protein and fat general energy was, in which the more protein was found in the mantle howler monkeys, which was explaining their differentiation from the macaques, which had higher amounts of fat in their milk. So linking this nutritional profile of the milk to what the microbial community looked like. And then across all the 9 primate species, there were 7 bacterial strains that were shared. So almost every mother had these 7 bacterial strains, and they may play an important role in infant human health and immune development. And 2 of them, one strain was a Staphylococcus and 2 strains were Streptococcus. They were in really high relative abundance here, making up almost 50% of most of the communities, and they're also common in other human milk studies as well. So I think important taxa that we can focus on, and we are continuing to focus on. So then after that study was complete, we really wanted to start asking these questions. Well, what happens over time? We had some temporal sampling in that first study, but we wanted to really get a better resolution. And so we went into the milk repository, figured out which samples had the most resolution over time. So these are the moms, so 6 different moms, different babies, different facilities that had good temporal sampling. And so we found that there were some slight changes over lactation, so changes in the number of strains that were in, slight increases, slight decreases, but nothing incredibly pronounced. This is each mom is in a color with a species, and they really kind of stayed around in the same space. So their composition was shifting a little bit, but not anything dramatic. But one thing was really interesting from this study was the Streptococcus species was decreasing over time, and this was one of the Streptococcus species that we found that was in all of the 9 primate mothers. So we saw this slight decrease over time, suggesting that maybe the immune modulatory regulation of bringing this bacteria into the mammary gland may be reduced as the infant ages. And I'd be really interested if you could find this in the child studies and see what's going on with this there And so then we noticed that there was one gorilla, Anju, was at a different zoo than the other gorillas, and we kind of saw that there seemed to be a facility effect there. And so we then did our next study was looking at facility effects. So we had rhesus macaques that were housed with Japanese macaques, rhesus macaques that were housed with olive baboons, and pigtail macaques that were at another facility. And what we found, there were facility-linked changes to the milk microbiomes. So the rhesus macaques with the olive baboons were here in a cluster together, still species-level effects, but they were grouped within the facility. And then the rhesus macaques that were with the Japanese macaques had a facility signal. And we found that this was impacting some signals of microbial function. We predicted what these microbial functions were, and we saw some pathways of particular hormone biosynthesis was changed, suggesting that ways that people were managing these species at different facilities is impacting the microbial function and potentially the function of the offspring, the microbes in the offspring. Okay. So that's kind of a synthesis of what we did to date, and then I'll talk about the PNAS paper here in the last few minutes of my presentation. So we really had touched on what could be causing these selective pressures, and at most we were always able to maybe explain 50% of the variation with what we were putting in the model, with the host species, environment, the nutritional content, and some evidence of infant age having some effects, but somewhat mild. So we went big. We decided, let's go and find every single mammalian species which had at least 5 different individuals that had been sampled, at a roughly similar time, either early lactation or mature lactation. And here they all are. We spanned all 4 placental superorders. Within each superorder, we tried to capture multiple diet types, multiple environments, and also have multiple milk nutrient profiles as best we could. But those tend to be evolutionarily conserved more. And when we did this, we found that again, it is host diet, evolutionary history, environment, and nutritional content. They influence milk microbiomes across all mammals as they do within primates. I'm highlighting humans here. So humans weren't the lowest in microbial diversity here. Primarily marine carnivores certainly have some of the lowest diversity. They also nurse for the shortest part of time. Harp seals, I think, only nurse for a few days, and they have super fatty milk. Just some anecdotal things there. But we did see that you could see clustering by superorder, by whether they were in, for instance, marine wild animals were particularly unique. We saw that carnivores could separate from herbivores. Particularly herbivores had certainly some of the highest microbial richness. And that's also what we see in their guts. Herbivores typically have the highest gut microbial richness, so that is related to their milk. And we also saw that, the milk nutrients was playing a role here. And so pretty much each one of these things explained about 5% of variation in the composition. And we tried to figure out what was it about diet or the milk nutritional content. We found that how much plants you ate, how much fish you ate, and how much fruit you ate actually could explain some of these shifts in microbial composition, and that the amount of sugar in the milk was explaining some of these differences and why species had different microbiomes than others. And we tried to figure out, was it diet that was directly impacting these profiles of the microbiome, or was diet maybe acting on the nutritional content of the milk? Like what the mother eats would then impact her nutritional content, which would then impact the microbiome, or was it just what she was eating would directly impact the microbiome? So for species richness, we saw that diet was directly impacting microbial richness. So and with that being that herbivores had higher richness and higher richness in their milk, having some evidence for enteromammary trafficking. And then we found for the composition that diet was indirectly impacting composition through its impact on sugar, its direct impact on sugar. So I just have a few slides left. So what we saw here was that there was some selection. All of these things mattered that we had seen before, but really we could only explain about 20% of the variation across this very wide diversity of mammals. And so we started to think about, well, what about these other community assembly processes, these that may be more stochastic? And so we went to compare what milk microbiomes look like compared to mammalian gut and skin microbiomes. So we took these 2 different data sets that have hundreds of species in them also, or multiple species at least, and we subset these data sets to be similar species that we had. So 32 host species here, 31 host species, and we did these assembly models to figure out what were the community assembly processes. So you have selection, and then you have these just stochastic processes that can happen. And in milk, selection was only about 20%, 40% in the gut, and about 60% in the skin. So much higher selection in those 2 other environments. And then you kind of see this reverse with ecological drift, for instance. Ecological drift was very high in milk microbiomes compared to the others. And from ecological theory, it's predicted that when you have just lower species richness in general, whether it's a plant community, a bird community, a microbial community, you're more likely to experience ecological drift. And indeed, we saw that milk had lower microbial richness than the gut and the skin. So maybe it's just a process of just having low diversity. And it was really neat that in these 2 analyses, there are two independent analyses. We found that selection was acting on about 20% of the community assembly processes, and then we were able to explain 20% of the variation by maternal diet, host species, environment, and nutritional content. So it's like it seems that we've explained everything that we can about what is the selective pressures acting on these communities. So to just wrap up, humans compared to primates, we have the least diverse milk microbiome, but there's at least 7 bacterial strains that are shared, and I think those are really fascinating to me because they are conserved all across primates. Like it just seems to reason that they have to be serving some sort of function to maintain this conservation across such long evolutionary time. Evolutionary history, environment, nutritional content, and diet matter for all mammals. And I think the stochasticity, like we can predict parts of these communities, but I think we also have to accept that some of them are just inherently stochastic and we will never be able to explain them. And I think that that's likely a result of so many sources of microbial inoculation, that it's coming from multiple environments in the mom, multiple environments from the infant. So for us, for the work that we're doing at Suze and the potential to manipulate these milk microbiomes, we're really focusing on diet changes to the mother. We see this in gut microbiomes as well, and really thinking about, we have wolves that experience inflammatory bowel disease, we have cheetahs that are experiencing gastritis, and really doing a lot of studies to understand how to give them the proper diet that gives them the healthy gut microbiome that they can then nurse their offspring to have healthy microbiomes. We've thought a little bit about probiotics. The wildlife probiotics that they put in there, none of those species are actually found in any of the species that we have at our zoos that we've looked at. So they don't even seem like they're proper probiotics. We would have to develop our own. And we're in that process for rhinos. We're looking at what would be ideal probiotics for rhinos and how to seed that into infant formula for them. And then we've got a study with macaques where we're trying to look at how much of mom's milk is actually being seeded into the baby's gut. Okay, with that, I'll thank my collaborators and funding and take any questions. Sorry, I think I ran over a little bit.